For the first presentation, we have Dr. Sarkis Mesmanian. He's the co-founder of Axial and also sits on our board of directors. He received his PhD training in microbiology and immunology. He's also a professor of microbiology at Caltech. He's won numerous awards, just to name a couple, being named by Discover Magazine as one of the best brains in science under 40. He was recently received the MacArthur Foundation Genius Award. He's also a founder of 2 biotech companies and serves on the scientific advisory board of over a dozen companies, academic centers, and not-for-profit foundations. So we are very lucky to have him here today presenting to us, and we're excited to hear from him in just a minute. We also have Dr. Stu Campbell. Dr. Campbell joined Axial in 2017, and he serves as our CEO and head of research and development. Stu holds a PhD in organic chemistry. He brings over 25 years of experience leading and building teams with a strong track record of discovering and developing novel therapeutics for patients that need them. To that end, in previous roles, he's played an integral part in the discovery and development of 5 clinical stage drug candidates, which is very impressive. He's also a co-inventor on more than 20 issued patents. So again, we're very lucky to have Stu presenting to us and also leading the charge at Axial here today. So without further ado, I can now turn over the control to Sarkis. So let me stop sharing. And Sarkis, if you're able to load your slides, I think we'll be all set.
I should be able to. And are you able to see my slides now?
We are, thank you.
Okay, just give me 1 second to get this into the presentation mode, which, Jim, it should be in now.
Perfect.
And really thank you for allowing me to present our work today that has supported the clinical activities at Axial. And so just really briefly, just to add to Jim's background, I'm a basic scientist here at Caltech, a microbiologist by training, and I'll take you through some of the history of the technology and the basic science that went into the development of clinical candidates that are now being tested in the ASD population, as you know. And so by giving you the background of the fundamental science that was done that both identified targets and allowed the testing of particular molecules in animal models. And again, I'll only talk about mouse work today, but I think you'll see, based on the foundation that we've built in this preclinical work, that the technology that Axial has taken into the clinic has strong mechanism of action, at least again in the preclinical models, and it gives us hypotheses to test in humans. And I think we're taking questions only at the end, so I'll go through the presentation and then turn it over to Stu for his presentation, where, as you can imagine, he'll talk about the clinical work, and then we'll have time for Q&A at the end. So I'll give a little bit of background on the microbiome in general. So microbiome has become in vogue in the past decade or so, where we hear a lot about bacteria that live in and on us through a variety of different media and scientific outlets. And obviously the medical community has really become aware of the potential effects of the microbiome on human health. But again, a lot of the work that's been done to date has been in preclinical or animal models. And I think, Axial is one of those companies that's really bridging the gap between this solid foundational science and the application of that science to help patients. We have about 100 trillion bacteria that live in our bodies. Most of this is in the lower gastrointestinal tract, in the colon, and the numbers generally exceed that of human cells. So we have more bacterial cells in our body than human cells, which I think is still quite astounding. In terms of the genes, our microbiome, which is the collective genomes of our gut bacteria, has 150 times as many gene families as our own genome. Many people have referred to this as our second genome, because the two, our own genome and that the DNA of bacteria, together collaborate to mediate health and disease, again, in a variety of different contexts. And in terms of function, the microbiome, all the cells in our intestines have the metabolic capacity equal to the liver and about the same weight as the brain. Again, as I've emphasized, and probably will again, a lot of the work in the microbiome field has been done in animal models, but many of those animal models are of disease. And so we now know that there are links between the microbiome and a variety of neurological diseases. We'll talk obviously about autism spectrum disorder today, but the microbiome has been linked to other emotional behaviors, including anxiety and depression, as well as diseases, neurodegenerative diseases such as Parkinson's and Alzheimer's disease. Again, based on its metabolic capacity, the microbiome affects metabolism throughout the body. I'll actually give you an example of a particular molecule that enters the brains of mice. And so our entire bodies are bathed in these microbial molecules, and therefore the metabolism, the co-metabolism between bacteria and us mediates effects that have been linked to metabolic disorders such as diabetes and cardiovascular disease. And really a lot of work has gone into obesity. And of course, when we think about bacteria, we think about the immune system. It's a reciprocal relationship. Our immune systems exist to fight off viruses and bacteria, but our immune system also learns from colonization by symbiotic or beneficial organisms. In fact, many immunologic functions are bolstered or improved by the presence of particular bacteria. And there are a large body of research connecting bacteria to chronic inflammatory diseases such as IBD and gastrointestinal infections. Specifically thinking, excuse me, about the gut-brain connection, which again is the theme for today. A lot of different conduits and interactions between the gut and the brain. I think a lot of this may just be intuitive. We feel certain things throughout our daily lives that remind us about this connection, but on a molecular and cellular level, there's just a rich communication between the intestines and the brain. It happens in a number of modalities. I'll go fairly quickly through this because we could actually spend so much time just thinking about how these two tissues communicate. But there's a lot of evidence that particular hormones and neurotransmitters are produced in the gut, and that the production of these metabolites or molecules are mediated by gut bacteria. For example, about 90% of the serotonin in our bodies, 50% of the dopamine in our bodies are in our gastrointestinal tract, and their production is subject to regulation by the microbiome. About 70% of our immune cells are in our gastrointestinal tract at any one given point in time, and these immune cells are constantly receiving signals. Their phenotypes, their activation states are affected by the constituency of the microbiome. As I've already described, the microbiome makes many molecules of different classes: small molecules, lipids, proteins, carbohydrates. In fact, we don't even know the entire chemical space yet, despite a lot of effort. But suffice to say, there are many hundreds, if not thousands, of molecules produced by bacteria that enter our circulation and essentially go to all reaches of our body. And this is really going to be the focus of today's discussion. And then there's certainly neuronal connections. The vagus nerve is one, probably the most predominant highway for communication between the gut and the brain, as well as other peripheral organs that are connected to the brain. And so there are certainly mechanisms by which the nervous system can mediate this interaction. Focusing on ASD, again, there's been a broad set of studies to show that the microbiome affects a number of different behavioral outcomes. Obviously, as we know, autism is characterized by deficits in social communication, repetitive and restrictive behaviors. But in addition to these core features, it's clear that subsets of individuals with an autism diagnosis have other comorbidities or features. Gastrointestinal symptoms can range in the autism population from 30 to about 50 to 60%, depending on the studies. Again, this audience knows that this could manifest in a number of different forms of gastrointestinal symptoms. And of course, that initially caught our eye, that this neurodevelopmental disorder is coincident with an increase in GI symptoms. A large body of research showing immune dysfunction in ASD, as well as metabolic abnormalities, which are also well known. And as I just described, the immune system and the metabolic system are subject to regulation by the microbiome. So these were just hints early on in our research because of the GI issues and then the immune and metabolic issues, that perhaps the microbiome may play some role in autism. I won't talk too much about aggression irritability, but as we know, that's a feature common to many individuals with autism, as well as anxiety, which I will touch upon again in the mouse studies, but bear these two features in mind because they're going to be important for the clinical data that Stu will present. It's been an increase in the diagnosis of ASD. There's a debate in terms of what is mediating this increase that we've seen over the last few decades. Certainly, we're much more aware of autism now than we were a generation or two ago. There have been changes in diagnostic criteria, well known to this audience, that account for a large portion of the increase in ASD in the US and presumably in other geographies as well. But it also opens the possibility, and there's growing evidence for this, that there are environmental factors, whether they're directly causal or contributing or a consequence, all that remains to be seen, but there is missing genetics in ASD, and potentially that missing genetics may be explained by environmental features. Just quickly, again, these are statistics that we're familiar with, but many individuals worldwide and in developing nations, I think that we're still underestimating the prevalence of ASD. There's in addition to the social and medical burden, there's an economic burden of autism on societies. I've already mentioned these gastrointestinal issues, but there are about 24 studies more on the way showing that the microbiome is different in individuals with ASD than neurotypical controls. This is done by high throughput sequencing, where we can catalog the membership of the microbiome, the different species, the different genuses of bacteria that are found in the microbiome, and there seems to be a different representation, different organisms between ASD and healthy controls, and different levels of organisms that may even be shared between neurotypical individuals and ASD. So again, a lot of work has been done and more needs to be done to understand what those connections are, what are those organisms that may be hallmarks of autism. And I urge you to think about organisms that may be overrepresented in ASD, that may be pathogenic, but there's also a lot of evidence showing that certain organisms are missing in individuals with autism. These may be protective organisms, and so it offers the opportunity to supplement a deficient microbiome. And so our journey, again, I'm a microbiologist, and so our journey into autism began about 15 years ago now, through a collaboration with Paul Patterson. Paul Patterson was a faculty member here at Caltech, a neuroscientist, very interested in the role of environment on behavior. There's been a number of epidemiologic studies showing that an infection during pregnancy leads to an increased risk of autism. Large studies, epidemiologic studies from Sweden and Denmark inspired Paul to do the work I'm about to describe, but since then there's been additional studies here in the US and in other geographies really identifying this link between maternal infection, a very severe bacterial or viral infection associated with a fever during pregnancy that leads to an increase in autism. There's, as you can imagine, a lot of people now looking at the epidemiology of COVID and whether or not that's going to alter rates and diagnoses of autism. So Paul Patterson, again, a basic scientist, modeled this epidemiologic evidence in mice. So essentially gave pregnant mice the flu and showed that it recapitulated many of the features of ASD, which I'll show you in a second. And then subsequently, we started moving away from live influenza infections for technical reasons, for practical reasons. We now use a viral mimic called poly-IC. So this is going to be important for the talk. We essentially trick the mouse's immune system to thinking it's being infected by a virus by giving this chemical. Again, it's a widely used reagent in immunology to essentially activate the viral response in mice and other animals. And so what happens when you essentially induce the flu or activate the immune system in a pregnant animal? The offspring show a number of features that are associated with autism that you can see here, including sensory issues, not just the hallmarks of ASD, but also some very curious neuropathologies, some of which have been reproduced in post-mortem human brains of individuals with ASD. So this model, again, it's an epidemiological model, but it mirrors both the behavioral effects as well as the brain effects that have been shown in humans. So we think that it's quite a powerful model. And so several years ago, again, the culmination of the work that we did with Paul led to these studies. And so, as I mentioned, there are reports in humans that there are various different types of GI dysfunction or GI inflammation in ASD. So one of the first features that we looked at in this animal model is to ask, are there gastrointestinal deficiencies in mice that have this feature of autism? And so the animal models here these are the offspring of saline injected mothers. And so that's just a control. So we just give salt water as a control injection, and then P is for poly-IC, that viral mimic that I just mentioned. This assay looks at intestinal permeability by essentially giving a fluorescent dye orally to the animals and then looking in the serum for the levels of that dye. The more of this fluorescent dye we see in the serum, the more leakiness of the gut, so the more impaired the intestinal barrier is. And you can see that offspring of poly-IC treated animals have a greater level of leaky gut than saline control. So very nice to be able to reproduce this feature in mice, again, that has been shown in some individuals with ASD. In this study, we wanted to ask, is there a role for the microbiome in potentially preventing these gastrointestinal symptoms? So we treated the animals with Bacteroides fragilis. Bacteroides fragilis is a human symbiotic commensal organism. It's found in about 50% of the healthy human population. It has very potent abilities to repair the intestinal epithelium, and you can see that here. So these same poly-IC offspring, we're not treating the mothers, we're treating the offspring with this one probiotic, reduces the level of leakiness in the intestines. And then we probed in on a molecular level. I won't spend too much time on this. And again, this data is published in this 2013 paper. But the leakiness of the gut is mediated by specific proteins called tight junction proteins. So we went into the epithelial cells, which line the gut, and asked what are the levels of these proteins. And we see that poly-IC mice have a reduction in Claudin 8 compared to saline mice, and an increase in Claudin 15 compared to saline mice. This profile of reduced Claudin 8, increased Claudin 15 has been shown in animals to result in leaky gut. And so we, in other systems, so we reproduce that in this mouse model of autism. And again, as you can see, Bacteroides fragilis repairs this effect, meaning it restores the production of these tight junction proteins. And again, these are the proteins that form the seal between epithelial cells and mediate the integrity of the intestinal barrier. And so there are GI symptoms. There's more that I won't show just in terms of this background study. And again, this is all in the literature already. But of course, we wanted to look at the effects of behavior with probiotic treatment. First, we looked at anxiety. This is the open field test, where a mouse is put into an open arena. And the more timid or anxious the mouse is, the closer it stays to the walls. The more bold the mouse is, it enters the center arena. And then we can track the movement of the animals with the video with the video camera and tracking software. And so we looked at the number of times that animals enter into the center of this arena. And so you can see poly-IC offspring enter the center less than the saline controls. And then once they do enter the center, they spend less time there compared to the saline controls, meaning that these animals show features of anxiety. They stay closer to the walls compared to otherwise healthy behaving animals. And then again, the poly-IC animals that were treated with Bacteroides fragilis, they enter the center similar to the control animals, and they spend the same amount of time as the control animals. So we can essentially repair or restore anxiety-like features in this mouse model with a single probiotic. Same with vocalization. So here we're using an ultrasonic microphone to essentially eavesdrop on mice and listen to their conversations. Again, in ultrasonic vocalizations, we can look at the number of calls and the duration of each call. And again, you can see this pretty stark deficit in how often these animals communicate. And if they do communicate, their sentences, if you will, are shorter than the control animals. Again, this vocalization phenotype is completely restored by treating the test animals with an oral probiotic. So very gratifying that we can improve this core feature of autism, at least in mouse models. And a test for repetitive behavior is the marble bearing test, where we take the animals, line their bedding with marbles, and then score how many marbles they bury in a given amount of time. And you can see that control animals bury about 30% of the marbles in this 10-minute testing period, whereas the poly-C offspring will bury over 40% of those marbles, again, a feature that's corrected with Bacteroides fragilis treatment. And so, I provide this background both in terms of GI and behavioral symptoms, just to show you that the microbiome can affect what are endpoints mediated by brain activity. So we wanted to then ask, what is that mechanism of communication? Similar to what I alluded to in some of the background slides, how is the gut and the brain talking to each other in this animal model? We looked at the role of the immune system. We didn't see a huge effect there. But we did see an effect based on the production of microbial metabolites. And so we did what is called a metabolomics screen. So we looked in the serum of these animals and asked, what are differences in the chemical composition in their blood? What are the small molecule differences that we can catalog between these various different groups of mice? We found a number of different changes. I'm going to show you one, and we're going to focus on one metabolite. But again, bear in mind that there are a number of other small molecules which have similar profiles. And so the molecule that we focused on is a metabolite called 4-ethylphenyl sulfate, or abbreviated as 4-EPS. And we chose to study this molecule because there's over a 40-fold increase of this molecule in the circulation of animals that show behavioral outcomes relative to their control counterparts. And so this was the largest effect in this particular animal model, in this particular study. And so we chose to study this metabolite because we wanted to understand, is there a bioactive role for 4-EPS? And then I can show you that treatment of those same poly-C animals with Bacteroides fragilis, which, as you saw, improved GI and motor symptoms, reduces levels of this metabolite. So the initial hypothesis based on this data is that perhaps 4-EPS may be pathogenic or may be contributing to behavioral and GI effects. So what is 4-EPS? We're still learning the biology of this. When we discovered the metabolite, there was very little known in the literature about this molecule. But what we now know, and I'll show you more data, is that the dietary molecule tyrosine, which is an amino acid that is found in many, many foods that we eat, is converted to 4-ethylphenyl by gut bacteria. 4-ethylphenyl enters the circulation and is very rapidly sulfated to 4-EPS, or 4-ethylphenyl sulfate. Again, that happens in the liver. So essentially the effective molecule that mediates an effect is 4-EPS. And so I showed you the data in mice. We reproduced the elevation of 4-EPS in another mouse model of autism, a genetic mouse model of autism. So that was gratifying, but certainly we wanted to look at its effects in humans. And so last year we published a study where we looked at this metabolites in 129 individuals with ASD compared to 100 typically developing individuals. These are samples from the CHARGE study at UC Davis. We look in both plasma and feces, but it's focusing specifically on the plasma. Really heartening to show that the animal data did translate into humans, that there is this subset of individuals, about a third of the ASD population in this one particular study, which have very, very high levels of 4-EPS. In fact, they're the highest in those individuals that have gastrointestinal symptoms. So a correlation, certainly, but a really intriguing one. And this really emboldened us to now ask the next question: is 4-EPS potentially a biomarker of ASD? Maybe it's irrelevant to ASD pathology, or maybe it's contributing or even driving symptoms. And so we recently, just a couple of weeks ago, published this study which directly tested the biological effects of 4-EPS. And so again, as I mentioned, we had discovered 4-EPS, and so we had to go back now, and through a lot of work in collaboration with Michael Fischbach at Stanford, we identified the bacterial genes that can convert tyrosine to parecomaric acid to 4-vinylphenol to 4-ethylphenyl. Again, this is what the bacteria produce in the gut. And then ultimately this is sulfated, and the 4-EPS is the molecule we find in circulation. We engineered two different groups of bacteria to either produce 4-ethylphenyl or not produce 4-ethylphenyl. They're the exact same bacteria, except this group of bacteria contained the three genes. for this chemical transformation. And then these bacteria, again, same exact strains, but are missing the gene. So a very clean system comparing groups of bacteria that produce or do not produce 4-EP. And then we colonized mice with these bacteria. And so now we have groups of animals which have no 4-ethylphenyl sulfate in their bodies, and other groups of animals that do contain 4-ethylphenyl sulfate. And so again, otherwise everything is identical in these mice other than this one molecule. So a very simplified system, but a very clean and rigorous system. So we validated that indeed 4-EP is produced in the feces. 4-EPS is found in the circulation. This is the urine data. We have similar data in plasma. And we now show that 4-EPS enters the brains of mice because once it's produced in the gut and sulfate in the liver, we do find detectable levels in the brains of these animals. So we wanted to look at how the 4-EPS may affect the brain. And so we show through this imaging study where we're looking at activity levels in different brain regions that there are certain regions, most of which are in the limbic system, which are known to be mediate fear and anxiety responses, are activated by the association with 4-EPS. So what you're looking at in red are areas that are more active in the 4-EP positive group compared to the 4-EP negative group. And then there are other areas which are less active in the 4-EP positive group. So this work was done with Daniel Holschneider at USC. We've also, in collaboration with Daniel, looked at functional connectivity, and indeed that is affected as well. So different brain regions seem to not communicate similarly between these two different animal models, or between these two different groups that have either 4-EP or not. And so based on that general feature of changes in brain activity, we wanted to focus in on their particular cells and molecules that are mediating these changes. And so we worked with Daniel Geschwind at UCLA. And so Dan is a very well-known autism researcher, very much focused on the genetics and the effects of autism on brain activity and behavior. And so with Dan, we profiled 6 different brain regions of these animals, the 4-EP positive and 4-EP negative groups, and looked at gene expression. So again, we're looking at RNA. At this point, this is an RNA-seq experiment, and we showed that the vast majority of the genes that are dysregulated, this pie chart shows which locations show the most effects at the level of gene expression, are in the PVT, the paraventricular nucleus of the thalamus. And so when we zero in on the PVT and ask what cell types are most affected, we certainly see changes in neurons, but we were really struck by these changes in oligodendrocytes. And again, I'll get into this a little bit more, but you can see that there are very significant effects in newly formed oligodendrocytes and mature oligodendrocytes. So a number of genes mediate these 2 different subsets of oligodendrocytes, where oligodendrocyte precursor cells are immature, and then through a well-known developmental program, become mature oligodendrocytes. And so we looked specifically at the genes that are markers of oligodendrocyte precursor cells, and you can see those are all upregulated in the 4-EP positive mice, and then all of the genes that are mediating the features of mature oligodendrocytes are reduced in the 4-EP positive mice. And so we interpret this to mean that oligodendrocytes are not maturing in animals exposed to 4-EP. So potentially 4-EPS arrests or slows down the maturation of oligodendrocytes. So what oligodendrocytes do, among other things, but their primary function is to myelinate neurons. Myelin is the protective sheath around long neuronal axons, and essentially helps neuronal activity. And so again, these precursor or immature cells do not myelinate, whereas the mature oligodendrocytes do. And so based on the genetic signature, we wanted to go in and validate, indeed, do we see an arrest or a decrease in maturation of oligodendrocytes in 4-EPS containing animals? And in fact, we do. So if we look at the maturity coefficient of oligodendrocytes, there are less mature oligodendrocytes in the brains of 4-EP exposed mice compared to 4-EP negative animals. No total difference in oligodendrocytes. It's not as if the cells aren't there, they're just not maturing. And so just for the sake of time, I will go quickly, but again, this study was published a couple of weeks ago. We validated this feature by looking using a different technique called flow cytometry, and gave us the exact same effect, that in the presence of 4-EP, there are less mature oligodendrocytes. We can recapitulate all of this activity in vitro, and so we can take brain slices and bathe the tissue in 4-EPS and essentially show that just in a completely cell culture system, 4-EPS has the same effects that it does in mice, that it reduces oligodendrocyte maturation, and it reduces the co-localization of oligodendrocytes and neurons with each other. And so this doesn't prove, but I think it strongly suggests that 4-EPS that gets into the brains directly acts either on oligodendrocytes or neurons. That's our working hypothesis. And so again, since oligodendrocytes myelinate neurons and mediate, and the myelination of neurons we know affects different types of behaviors, we wanted to look at myelination patterns. So looking at actual cells through cryo-electron tomography. These are the cells in the corpus callosum of mice we're looking at directly, and we can measure how many of those axons or what portion of those axons are myelinated. So we see this increase in unmyelinated axons in the 4-EP positive group. So essentially there's just less overall myelin being laid down in this tissue. And then when we measure the thickness of myelin using a G ratio, which accounts for the irregularities of the cell types, a higher G ratio means thinner myelin. And you can see when we look across many, many dozens of neurons that there's a decrease in the thickness of myelin. And then we can just directly measure the thickness of myelin, and you see that that's reduced in the 4-EP positive group. We wanted to look specifically at the paraventricular nucleus of the thalamus, or the PVT, because of this stark gene expression data that I showed you. And indeed, we see more disorganized myelin in the 4-EP positive group compared to the 4-EP negative group in the paraventricular nucleus of the thalamus. This is DTI studies. And so again, both of these, both looking at myelination as well as disorganization of myelin, both give us the same outcome, that because of the reduction in oligodendrocyte maturation, there's a change in myelination or white matter patterns in the brains of these mice. And so ultimately, we want to correlate this to behavior. We want to, again, ask that central question. How is 4-EPS inducing changes in the brain? And then importantly, all the changes in the brain manifesting into behavioral outcomes. And so we behavior tested these animals. And so I've introduced the open field test to you, and you can see mice that were exposed to 4-EP. So the mice that have 4-EPS in their brains and have less myelin in their brains have this anxiety phenotype. They enter the center less compared to the 4-EP positive group. No change in distance movement. This is a metric for essentially health. It's not as if the animals are sick because of the 4-EP and just huddled in the corner and not entering the center. These mice are perfectly healthy. Again, we've looked at a number of different features of these animals. There's nothing toxic about 4-EPS. It does appear to affect decision-making in the animal. We can reproduce the open field test with another anxiety test called the elevated plus maze, where the more anxious the mouse is, the more time it'll spend in the closed arms of this elevated maze is about 3 feet off the ground. And then the more bold or adventurous the animal is, the more time it'll spend in the open arms and then get to the terminus. And you can see that the time spent at the open end of the terminus of the open arm is greatly reduced in the 4-EP positive group compared to the 4-EP negative group. So really strong data that this metabolite appears to be causing anxiety-like behaviors. We looked at social interaction as a core feature of autism. There's a trend to reduced socialization between bacteria, and this peculiar change in anogenital sniffing. Essentially, these animals are socializing differently if they were exposed to 4-EP than not. And when we look at ultrasonic vocalization, again, as a surrogate for communication, the 4-EP positive animals communicate less than the 4-EP negative group. So it looks like there are features of these animals that also mirror hallmark features of human autism. And then finally, just bringing this back to the oligodendrocytes, a lot is happening in these animals. We looked at oligodendrocytes, we found the effect on behavior, so we wanted to link those two together. And so what we did is we treated the animals with a drug called clemistine fumarate. So this is purely a research tool. At this point, it's not part of the clinical study. But this research tool we know because clemizine fumarate essentially forces or drives oligodendrocyte maturation. This is the data I've shown you here. There's less mature oligodendrocytes in the 4EP positive group, but when we treat with the drug, you see that the less mature oligodendrocytes recover their phenotype. Okay, so even in the presence of 4EP, we can force these oligodendrocytes to mature. And if that happens, we can overcome the behavioral deficits in the 4EP positive mice. So looking at the elevated plus maze, again, this is the data I showed you, but when we treat with clemizine fumarate, the mice will go into the open arms more than without the drug. The drug improves the time that the mice spend in the center of the open field and also reduces this marble bearing test. So again, this is from a research standpoint, really identifies oligodendrocytes as a potential target in this context for the effects of a microbial metabolite and its ability, oligodendrocytes' ability to mediate behavioral outcomes. And so I'll conclude with where the biology stands today is that changes in the microbiome of ASD also change the metabolome, meaning that it changes the small molecule production from the organisms in our gut. Again, this happens in ASD, it happens in a number of different disorders, but in this particular context, the effects on the gut epithelial barrier, the compromise of the intestinal lining leads to an influx of metabolites into the circulation. 4EP being one of those metabolites is sulfated in the liver. This effectively results in an increase in its concentration in the circulation in the brain and ultimately mediates the arrest of maturation of oligodendrocytes, keeping them in this immature or precursor state, which then results in less myelination throughout the brain and changes in behavior. And so the concept, again, this is a springboard to Stu's discussion, but the concept, I think that these studies in animals embolden is the ability to potentially drug diseases like autism through the gut and not try to get drugs into the brain, which we know is challenging and is more fraught with side effects. If we can directly target therapeutics to the gut and to the gut microbiome, we may be able to get effective treatments that don't have the side effects of molecules of drugs that actually target brain cells. And so I will just conclude very quickly to thank the people who did the work. A lot of the work that I showed you today was performed by Brittany Needham. She's a postdoctoral fellow in our laboratory. Our work is highly collaborative. We work with a number of individuals in academia, and obviously the team at Axial, which we work closely with, and these are our funding sources. And so I will thank you for your time and turn this Zoom over to Stu.
So thank you, Sarkis, for that. And I think what we have here is a really strong foundation. There's still a lot of things we need to work through and understand, and there's a lot of great research ahead of us. But what we want to do is really take work like Sarkis just went through and really adapt it and leverage it to develop new therapeutics, as Sarkis alluded to. And so our mission here at Axial is to really change the way we treat neurological conditions by exploiting how that gut microbiome that Sarkis talks about interacts with the human host. And so we are very squarely founded in Sarkis's work, both in autism, but we also have other work going on in other areas that are based on work that originated in Sarkis's lab and more recently in our own labs here at Axial. And I think what distinguishes us, at least on one level, from other groups or companies that focus on the microbiome as an influencer of health and disease is rather than take a probiotic approach, which may work well, and we're cheering for those people who are working on those types of things, but we want to look at the gut microbiome and dig deep to understand how the microbiome is influencing the host, whether it be in behavior or neurodegenerative diseases, et cetera, so that we can develop, I guess, more traditional looking drugs in the sense that they're small molecules, but ours are designed to work exclusively in the gut. So our drugs will be taken by mouth and they will travel through the GI system and they will never enter the peripheral tissues, at least not to any significant extent. And so just to recap very quickly what we're talking about here, the way we think about this microbiome gut-brain axis that Sarkis talked about at the beginning of his talk, we really think of three avenues of communication, major avenues. And Sarkis talked about the vagus nerve, and that's really the main trunk of the enteric nervous system. And so if I get my pointer out here, we know that certain things can travel from the gut through the enteric nervous system, get to the brain. And we're actually pursuing an intervention in Parkinson's disease based on this route of communication. We also have, as Sarkis mentioned, the gut microbiome can tune, educate, influence the immune system, and that can be leveraged in many ways for a variety of diseases. But where we're focused is really on these gut metabolites or gut small molecules that principally come from the microbes in the gut that enter peripheral circulation, get to the brain, and can impact things like behavior. And so this is our avenue into autism. And as Sarkis mentioned, the epidemiology and the prevalence of autism is increasing. I think most of the people here will appreciate this from a statistical point of view, but also just say, you know, mine is one of those 1 in 44, and I really want to do something that helps them become their full selves. So the really, I think, unfortunate thing is that there are only 2 drugs approved in the US that have autism in their approved label. And those are 2 atypical antipsychotics, Abilify or aripiprazole and risperidone, which I'm sure that many of the people on the phone or on the call here are familiar with those. They obviously have some issues, but we really have an unmet need here to help people with autism, you know, manage things like irritability and anxiety that these antipsychotics are meant to help with. But they create a lot of other issues while they do address these irritability and anxiety challenges. And so our motivation here and rationale is really what Sarkis just went through, is that there's a huge unmet need. The prevalence of GI disturbances and issues with people with autism, and that implicates that maybe the gut microbiome may be involved. And I think you've seen a wonderful story of how we can take a very rigorous scientific approach to understanding that relationship between the gut and autism. And so what we've got to, where Sarkis left off, was that we have this mechanistic hypothesis that there are these neuroactive metabolites coming from the gut that get to the brain and contribute to autism like behaviors and associated co-occurring conditions through a potential mechanism that involves myelination changes in the brain. And so this really leads to several different ways we could imagine intervening. And we'll talk about our first step at developing an intervention based on removing these microbial metabolites from the GI tract. But just to recap, we focus on things like 4-ethylphenyl sulfate that Sarkis just talked about. This is a little bit of the data he showed. And basically what he showed was not only is 4-EPS elevated in mice in that mouse model he talked about, but it also, when injected into wild type mice, induces an anxiety phenotype. And so it shows that molecules like 4-EPS that come exclusively from gut microbes do have pharmacology that's potentially relevant to things like autism. And so again, we worked with Sarkis and several others to publish this paper, which showed that in kids with autism versus matched controls, 4-EPS was elevated in a significant subset of the autism population. and it was associated with increased GI symptoms. The big question here is whether 4-EPS has the similar type of pharmacological activity in people that was shown in mice. And so the way we look at this as a drug developer is it's not in any way are we suggesting that 4-EPS causes autism. It's certainly not the case. But what we know and is that 4-EPS can contribute or exacerbate some of the behaviors, at least in mice. And the question was, could that also be true in humans? And it really led us to focus on not just 4-ethylphenol and its metabolite that shows up in the blood, 4-EPS, but a whole family of these metabolites, like paracresol, like indoxil, that also go through a similar process in the liver, where the liver sees them as kind of foreign substances and tries to get rid of them by putting things like sulfate groups on them. And it turns out that it's this class of metabolite, these sulfated metabolites like 4-EPS, paracresol sulfate, indoxil sulfate, that we believe are contributing in some way to exacerbating some of the challenges that people with autism have. And so what we have is this mouse data that Sarkis talked about, where chronically exposing to something like 4-EPS through a gut-mediated route does lead to behavior changes and also to changes in a lot of brain function, brain chemistry, that would lead us to think that we're onto something important here. And so the question really is, how do we intervene and test whether removal of these metabolites could actually help somebody with autism? And so we thought of several different ways along this route where one could intervene. You could change your diet. That may work to some extent, but it's very hard to predict how it will work in an individual because everybody's microbiome is different and everybody responds differently to these types of changes in diet. So what we thought was, let's take a very simple approach. Regardless of who the microbes are that are producing these metabolites, let's just remove them after they're produced, but let's remove them at the gut level where they're produced. And so we thought of one way to do this would be to adsorb them or sequester them or sponge them from the GI tract through a large molecule. And so we had this set of criteria. We said it needs to be a safe molecule that is tailored specifically to bind things like these microbial metabolites in the gut, and also can be taken chronically, and it is something that we think would both act in the gut, but also not enter the other tissues. So it won't enter the blood, it won't enter the GI tissues and so on. It will just travel through the gut as sort of an empty train that picks up these passengers along the way. And you'd simply pass them in your stool, and the net result would be a reduction in these metabolite levels in the blood and presumably in the brain. So when searching for a molecule that did this, safety was one of our key criteria here, because the one thing that when you want to give somebody with autism a medication, one of the top concerns, apart from will it work or not, is what else am I going to put this child through as a result of the medication? So safety was a paramount importance and is consistently to this day for us. So we looked high and low, and we actually found a molecule we call AB2004. And what it is it is a microsphere material that is a scaffold or a mold that is designed to sequester molecules like 4-EP in the gut. So these are less than 1 millimeter in diameter, but you can see they're perfectly spherical, which is important for chronic dosing. And the way it's made is they take a kind of it's a very highly engineered process where they create these spheres with a dopant small molecule. So they kind of imprint and build the sphere around these small molecules that look like the ones we're trying to remove. And then in the next step of the process, these small molecules, which are harmless, are extracted away, and it leaves a hard sphere with essentially pores or binding sites that are ideally suited to bind the types of metabolites that we're trying to remove. So when this enters the gut, it travels through the gut, it picks up these metabolites in the gut, and this is what's passed in the stool. So the material looks like this. It happens to be made of an engineered carbon lattice, and it is got very high surface area and these pores and binding sites that kind of are the mirror image of the material we're trying to remove. So now you've got kind of like putting pegs into a hole, essentially. And so this gives it some really, really important properties. And one might immediately go, well, gee, this looks like activated charcoal. It's not. It's anything but activated charcoal. If you compare this nice, smooth, perfectly spherical microspheres to the jagged particles, irregular ones of activated charcoal, you see that you can imagine taking this chronically, no GI irritation. Here, jagged edges, they're going to irritate the gut over time. So, chronic dosing could become a problem. It does not, it's because of their size, they're micro like around, half a millimeter in size, they're not going to cause constipation. You're not going to get black stools and so on. So there's a lot of advantages to this engineered microspherical lattice. And the other thing is activated charcoal takes out everything. It just wipes out all kinds of stuff in your GI. But because of the nature of these binding sites and pores, we've really restricted the breadth of things that this can sequester. Again, with safety in mind, we're limiting the types of things that are absorbed by the drug by building essentially a framework around the molecules that we want to absorb. And so we took this molecule back into the mouse model that Sarkis talked about. And what we showed was that treatment of these mice that chronically produce 4-EP in the gut and 4-EPS in circulation, we see that 2004 was able to significantly reduce the amount of 4-EPS in the urine. We also saw the same in the blood. And so that told us that this whole concept of pulling things out at the gut level does actually allow us to reduce these levels in circulation. And we saw corrections of these types of behaviors that Sarkis had shown were inducible by 4-EPS. We were able to correct these anxiety-like behaviors and repetitive behaviors in mice, in those same kinds of mice. And this is a separate paper that just came out at the same time as the Nature paper that Sarkis talked about, where this was published in Nature Medicine a couple of weeks ago. So with this preclinical data, we went into an open label safety and tolerability study with AB 2004 in teenagers with autism and GI symptoms. And it was really designed to establish that if we are going to go into giving kids with autism a medication, we really need to understand how safe and tolerable it is. And so it was a dose escalation study where it was 8 weeks of treatment, but at week 2 and week 4, the dose was escalated based on passing a safety set of safety criteria. And then at the same time, we took several measurements along the way to really understand whether the drug was able to reduce the bacterial metabolite levels in the blood and in the urine. So we could say, is the drug at least doing what it's supposed to do in the GI tract? And does that result in lower levels in the system? And then the third major thing we did was we said, look, there's a lot of data that Sarkis showed in mice, but that despite the breadth and depth of those behaviors, it's not always clear how a behavior in a mouse might translate to something you would see in a human being. So rather than narrowly guess that we might see an anxiety change or something like that, we measured a really wide range of behaviors: adaptive behavior through the Vineland, repetitive behavior, anxiety through the pediatric anxiety scale, and aberrant behaviors like irritability through the aberrant behavior checklist. And so what I'd like to do is take a couple of minutes to walk you through the outcomes of this trial. And so first off, we met our primary goal, which was to show that it was safe and well tolerated. The AB 2004, very safe. We had 2 serious adverse events, neither of which was related to the study drug. These are teenagers, they're rambunctious and active. So one of the serious adverse events was one of the kids broke his arm when he fell. Another kid got mononucleosis the day before he was supposed to start on the drug. So clearly not related to the drug. Everything else was temporary, mild to moderate, and was self-resolving, and usually around the GI system. So some modest GI effects, not in everybody, and were very minor. So no weight gain, no senescence, none of the neurological issues like tardive dyskinesia or something that you would see on the atypical antipsychotics. Absolutely none of those side effects. So we think now, the very beginning, we have something that at least is safe and well tolerated, and the kids had no trouble taking the drug. Because it's a powder, they simply mix it with food and eat the food. It has no taste, no smell. There's a little bit of a texture to it, but we had north of 95% adherence with the therapy during the trial. So we were really happy to see that at least it met those basic criteria. The question was, did it reduce the metabolites, and did it actually improve behavior? And I want to keep reminding everybody, this is an open label study, and I'll talk about that in a little more detail in a moment. But if we look at the entire cohort, which was 30 kids, I think this is 26 of those who had full measurements at the end of the study. When we looked at anxiety, and you remember that in the mice, anxiety was one of the major types of behaviors that we saw from 4EPS. And we saw maybe a trend to reduction when we looked at the entire cohort. But remember, we did not limit who could come in the study other than they had to be confirmed diagnosis of autism and have some evidence of GI symptoms. They could have high anxiety, low anxiety, didn't matter. And so we got the full spectrum. But when we did a post hoc analysis and pulled out the group that started the study at baseline, which is BL here, with high anxiety, and that on this pediatric anxiety rating scale is defined, not by us, but by the instrument as a score of 10 or higher, we saw a very significant improvement in anxiety over the course of the 8 weeks. And that reduction in anxiety score persisted through the 4-week recovery period after which, during which the drug was taken away. And what's interesting here and very gratifying is that the metabolites of interest, and we show here 4-EPS that Sarkis talked so much about, and the others like paracresol sulfate and indoxyl sulfate that I talked about, there's a whole family of these that we're looking at. They were significantly reduced by the drug. And what's interesting is in the subgroup with anxiety, we saw a different pattern of change of metabolites. So this may hold the promise to us being able to stratify people based on their metabolite profile. That's something we're still working on. We don't have all those answers yet because the data set's still pretty small, but it's something that really encourages us to go and keep collecting this data so that maybe we can understand in advance who will be more likely to respond to our treatment. The next thing was we saw a significant improvement in irritability. And so even at the full cohort level, where we saw a full spectrum of irritability scores coming in, we saw a significant reduction. And then when we did this same kind of post hoc analysis of the high irritability subgroup, which is anyone with a score of 15 or higher on this scale, we saw an even more significant improvement in some cases, normalization of the irritability score. And the key metabolites also were reduced by the drug. So again, we'll come back to this, was an open label study. So that means there's no placebo group in this study. And one might say, well, you're measuring behaviors in a subjective way in a population that sometimes struggles to communicate. So how do you know this isn't just a massive placebo effect? Well, the short answer is we don't really know yet, but the degree of change in this score is much higher than any placebo response in an interventional trial that has been published before, which means that there is very likely a residual treatment effect that's worth pursuing. And so on that basis, we said, if the irritability score is real, does that present an avenue to develop this drug? What's interesting is when we took the drug away, so now this is the same data that was on the previous slide, but we've added a recovery period that not only do we see within 4 weeks of stopping the drug, the irritability scores started to rebound. They didn't quite get all the way back to baseline, but they started to rebound. We actually saw all the metabolites go back to baseline within that same month. So all of this is consistent with, we reduce these metabolites, behavior improves, we take the drug away, metabolites come back, and behavior starts to revert back. So it really supports our contention that it's the drug that's doing this and not some entire placebo effect that we're seeing. That contention notwithstanding, I'll skip this slide, the only way to prove this is to do a proper gold standard randomized double-blind placebo-controlled study, which is what we're doing now. And in this one, we have a placebo group, a low-dose group, and a high-dose group of AB 2004 in the same population that we were looking at in the first study, except that we're now allowing kids in who have an irritability score of 18 or higher, which is exactly the same criterion that the aripiprazole and risperidone studies used. The trial design is very similar to what the aripiprazole and risperidone studies used to show that their drugs improved irritability. We've had this conversation with FDA. We've got our IND approved, and that has allowed us to go into this study with the idea of putting our drug 2 different doses against placebo. And so our primary outcome here is a change in irritability at 8 weeks, high dose versus placebo. Secondary endpoints include low dose against placebo and several other behavioral measurements as well. We also want to understand changes in metabolite levels. So we're measuring, taking several samples along the way, mostly urine, which is much easier to get from somebody with autism, but there are also a couple of blood draws in there as well to understand what's happening in plasma. And so the key inclusion-exclusion criteria for this study, obviously a confirmed diagnosis of autism, a couple of ways to do that, an irritability score via the ABCI of 18 or higher at screening, but we measure it multiple times at the beginning of the study. But it's the screening visit that we rely on to allow people to randomize into the study. But the irritability score is one thing, that's a parent-reported outcome. What we don't want to do is remove the clinician's opinion from this. And so it's very important for us to have the clinician's global impression, the severity score, as well as a guide to say, you know, in the clinician's opinion, this person has, you know, some issues and they score on the severity side of 4 or more on the Likert scale. Some evidence of GI symptoms is also a requirement. And then we're enrolling males and female adolescents 13 to 17 years old. We thought about going younger. We discussed this with the FDA and they said, look, do the teenagers first. If you corroborate your safety data from the first study and you present a strong case for safety, then you can go into kids as young as 5 years old. And so that's our plan. Key exclusion criteria, obviously, since we are targeting something in the microbiome, the metabolites that are produced by bacteria, anyone using antibiotics, that could be a strong confounder of our data. So those are prohibited. No oral controlled or extended release medication. Some kids are on extended release ADHD medications, for example, so those are precluded. We do have a way to allow kids to switch back to immediate release medications and get stable on those if they want to enter the study, but that's done on a per case basis with the clinician advising. So current use of antipsychotics, so we currently do not include anyone who has aripiprazole or risperidone in their regimen of medications. And if they haven't responded to the atypical antipsychotics before, they also are excluded from the study. Where is the study being conducted? We have, I think, 20 sites active at the moment, 18 in the US shown here, and one in Brisbane, Australia at Queensland Children's Hospital, and one in Auckland, New Zealand. And so these are all sites that are actively recruiting. You can find more information about this study at clinicaltrials.gov. This is our number, the NCT number, and you can find a lot more information about the study there. Or you could go to our study trial website, and we've called this the Tapestry Study. And you can find it at theautismstudy.com, and you can work through a set of questions and learn a lot more about what we're asking of people who come into this study and what we're trying to show. And we encourage you to go there and learn more about the study so that you can help us figure out, beyond the safety and tolerability of this drug, does it actually help kids with irritability? We think irritability is not the end of the story here. We obviously could go into anxiety and so on, but because the FDA has seen drugs for irritability before, there's some familiarity there, and it really de-risks the program from our standpoint. It's new biology we're trying to leverage. So to take the risks of the drug and its safety and the development path off the table, it really allows us to focus on proving whether this new biology could actually be leveraged for new therapies. And so maybe I'll stop there and thank the crew who allowed us to get this study done. And it's just been a real pleasure to work with everybody, globally on this study. And I think we're onto something very important here, and we need to prove this in this new study that's actively recruiting.
So thanks, Stu. Thank you, Sarkis. If you guys are okay, there are quite a few questions in the chat. I can go ahead and read those off, and we can try and get some questions answered.
Sure.
So we have one from Sarah. She says she's curious to see how our work is aligning with or complementing work being done at ASU on microbiota fecal transplant and/or their customized probiotics. Does your work address the same thing, or do you feel different goals and hoped for results?
Sure. Sarkis, you want to take the first stab at that?
Sure. So I think there are parallel approaches to try and address the same thing, right? Is if there are changes in the microbiome at the species level that results in a change in the function of the microbiome, and by function we mean the molecular output, many of the molecules that we showed you here. And so the approach taken by Jim Adams and Rosa Kraft-Melnick-Brown at ASU, who are colleagues of ours, and as Stu mentioned, we are rooting for everyone in this field. We just want to help the families and individuals. Their approach is essentially a wholesale change of the microbiome. So swap out the ASD microbiome for a healthy microbiome. At least that's the intended goal, to alleviate these effects. Ours, again, is an orthogonal approach where we are more refined in the targets instead of just an overall change in the microbiome. We're going after those molecules which may be mediating some of those negative effects through the sequestration approach that Stu mentioned. Again, and one can think of a number of other potential interventions which would achieve the same goals as well. We can talk about the trade-offs in terms of safety, in terms of efficacy. But again, I think, fecal transplants have been shown to be, quite safe in many, many thousands of individuals outside of ASD. So fecal transplants have been used for a number of other indications. And so, I'm personally bullish on their approach. And we hope that ultimately we both can help this patient population.
Yeah, I totally agree. We're really rooting for them, and it is different. They could coexist at some point, right? These two therapies. So, yeah, we're just excited to see where they're going to come out.
Okay, thanks, and great question. There are also quite a few questions from various folks just about the ages for the study. So as Stu outlined, our current trial is for ages 12 to just 1 day shy of 18 in terms of recruitment, and the plan is to move at some point into ages 5 to 18. But the question sort of everyone is asking is, do you know we'll be opening this up to young adults? And is that a plan for the future?
Yeah, gosh, this was a hotly debated topic inside of Axial, is where do we start? Because no matter where we start, we're leaving groups of patients who need this kind of in the lurch for a little while, and that's not our intent. We have to start somewhere, and we have every reason to believe that because of the mechanistic work that Sarkis showed, if that is at least a big part of what's going on here with these metabolites and a drug like 2004, changes in myelination are a very dynamic process that happens throughout life. Every time we learn something, every time we experience something, connections between different regions of the brain and neurons and so on are being made, strengthened, weakened, and all this requires what we call myelin plasticity, or the ability to rapidly change myelination. And so we believe that this has potential to work in adults as well. And I know, Sarah, you had mentioned that you have a 21-year-old who would be not eligible due to his age for the current study, but we plan to go into adults at some point, even as we go into younger kids as well. So we think it could be, we just don't know how well it might work in one group versus another, but we're going to have to just generate the data in a controlled clinical fashion to understand that and understand how useful it might be. So I hope that that's helpful. Maybe not a satisfactory answer for today, but I think it may help.
Thanks, Stu.
Give a sense of where we're thinking.
Sure. So the next question from Sarah is, are we seeing any comorbidity between the autistic gut and seizures, epilepsy?
Sarkis, do you want to take that one?
This is a clinical, I can start, but it's a clinical question, right, Stu?
Yeah.
Yeah, I'll just comment on the mouse models. None of the animal models that we've used have shown seizures overtly. In animals, oftentimes you have to induce the seizures. They don't happen naturalistically, and we haven't done those experiments. So at least on the animal side, we have essentially no data on whether or not seizures are involved in the process.
Yeah, we do know that there are differences, I should say, in the microbiome profiles of people who have epilepsy versus those who don't. And the current thinking there is that it's maybe a related field to what we're in the sense that it may be due to microbial metabolites, but in the case of epilepsy, it may be that it could be related to those neurotransmitters and those levels that are altered or controlled by the gut microbiome. And there's actually people working on that, not us at the moment, but there are other people working on that because they do see this connection and they want to see if it's exploitable in much the same way that we're trying to exploit the metabolites that we're interested in. And so I see the next question here.
If I may, Stu, I have a question for you. It's an 8-week trial, so it's short.
Yeah.
But I suspect, I could be wrong, more people on the call would know better than I do, that the behavioral instruments would pick up seizures, right? But
Not explicitly,
okay.
right? If it manifests in some other way, we might pick it up like a consequence of a seizure, but not seizures directly. They'll obviously be recorded as events that happen, and they'll be cataloged accordingly, but we're not explicitly measuring seizures in our study.
Yeah, so we'd be able to collect the data.
Yeah.
But again, I think the small numbers plus the short time frame of the study would suggest that we may not see those episodes. Individuals who have chronic seizures, I don't know if they would have entered the study in the first place. And so I think that those are considerations where, again, I think on the clinical side, it's going to be hard for, at least the way this study is designed, to address that.
And exactly. And so that leads, I guess, to the next logical question that Sarah asked here, which is, do kids need to stay away from seizure meds? And the answer in short is no, they don't need to stay away from them. If they take controlled release or extended release versions of those medications, then they would need to stay away from those. But in principle, the way that we would manage that is kids would take their seizure medications first in the morning, perhaps, and an hour later they would take the AB 2004, and that way we would avoid any potential impact of one drug on the other.
Okay. Thanks, Stu. Next question: what are microspheres made from? Does it also mop vitamins and minerals?
Yeah, so it's made from natural carbon product. So it's sort of a organic material that is baked essentially down into its basic carbon component, and it's built up around these small molecules to create the pores from there. And so does it mop up vitamins and minerals? If they share a chemical similarity to some of the metabolites that we're trying to remove, it could. But we obviously watch that and we look for things in the blood while the trial's going on from a safety point of view. For example, the vitamin B class is probably not impacted by those, and the vitamin D class is probably not. But we can't say en masse none of the vitamins and minerals are not minerals. It wouldn't impact minerals per se, but some of the vitamins could be. But we watch for that, and so it hasn't been an issue up to now. But we need to do the bigger studies to verify that.
Sure. Thank you, Stu. Next question: do you think we'll have any sites in Europe in the future?
Yeah, we have to. We've not gone to Europe yet. So we're doing this current study. We probably will not go into Europe in this current study, but in the next round of trials, we will absolutely be in Europe for sure. There's no reasonable path forward for approval for this drug without doing appropriate studies in Europe.
Yep. Okay. So from Shan, was wondering if it would be suitable for ASD kids who have high histamine levels, mast cell activation.
I don't think they would be precluded from taking the drug, but because of the way the drug is operating, we don't think it's going to alter somebody's allergy state or immune cell or immune state directly. So I don't know that it would, I doubt very much it would address anything related to an allergy condition. But certainly they could come in if they have irritability or anxiety, something like that.
Sure. And looks like the last question from Felicia is they have parents who are interested and wondering why the use of antipsychotic are excluded for the study.
It's a good question. And again, a hotly debated topic when we were designing this study. We actually had a couple of kids in the first study that I talked about who are on risperidone, and they seem to respond to this drug. At this stage of development, it's really important that we try to isolate variables essentially in such a way that we get a very clear answer whether the drug is going to work or not. And so to do that, one of the variables that we can control is the types of drugs that people take, for example, or the population that comes in. And we were a little bit concerned that antipsychotic use may confound the data, in which case, in order to see the effect of the actual drug, 2004, we might need a much bigger study to account for that potential confounding variable. So we decided, on balance, in this study, we probably should exclude those on antipsychotics. Again, probably not a very satisfying answer, but that's kind of the way we went through the decision.
Sure. Thanks, Stu. So question from Jacqueline. Her son has major GI issues, also completely nonverbal. Would this disqualify him from the trial? And then would the use of certain any GI drugs disqualify him for the trial? He's not on risperidone or Abilify. So no disqualifications there. I would just say if the GI drugs are extended release, then we'd have to take a look at that. But the nonverbal piece is not an issue in terms of
right. What we ask is that the kids are on a stable drug regimen for at least 30 days before entering the study, and they maintain that throughout the study.
Sure. Just a couple more comments have come through. I'm seeing if there are any last questions.
Yeah, the phenol and sulfate question is interesting. We talked about these metabolites as coming, like 4-EPS comes exclusively from gut microbes. Paracresol sulfate primarily comes from gut microbes, but there are other places in the diet that it could come from. And so we want to be careful to say it's not just these specific bacterial derived metabolites, but it could be more gut metabolites that are in this class. That so yes, that's a good point.
Okay. Does
I have a question about the exclusion criteria. If I understand correctly, the age range is strictly adolescent.
Yes.
Those who are currently taking an antipsychotic are excluded. Those who previously had an antipsychotic failure are excluded. And they have to have an irritability score above 18 on the irritability subscale of the ABC. Okay, so clinical scenario. Those who have a high irritability problem are going to have tried an antipsychotic in the past,
yes,
before they get to adolescence. Okay.
Yes.
Now, if it worked, then they will continue it and their irritability score will be below 18 and they won't be willing to stop it to be in the study.
Right.
If it did not work, then they're excluded based on that. So who's left to enter the study? And if you find a few brave souls who are willing to stop their antipsychotic and go back up to the qualifying score on the ABC, will they be representative of the autistic population?
Two great questions, Dr. Arnold. And I think that so who's left there? I think there are many people, at least that's what our clinical PIs have been saying, and our KOLs to say, there are a lot of parents who won't even want to start the antipsychotics because of the side effects. And so that's really the group that we're primarily focused on. Your point about those who would have tried and failed, or maybe tried and responded, but then came off because of the side effects, is a population we need to take a hard look at with respect to this study. So I think it is something that we are currently debating about whether we've inappropriately excluded some people from this, because I think that your point is well taken. There are several reasons why somebody may stop an antipsychotic treatment, and it may not be failure to respond. It may be the net effect risk benefit is not right for them.
Okay, it looks like we have two last questions, and I want to be cognizant of time. I know we're a few minutes over time, so Stu, if you still have a couple minutes, we can take these last couple questions. Okay. The stable GI regimen for 30 days includes some PRN medications, like day-to-day adjustments of constipation meds that are already a part of their plan.
Yes, it does. We can account for that. We know that, you know, to say stable drug regimen, that's going to fluctuate based on somebody's diarrhea or constipation. They're going to do it, take these drugs in an episodic fashion. But if it's part of their sort of history, then that's fine. Or acetaminophen for pain, right? These are things that are taken not chronically, not every day, but are taken sporadically. Those are fine. Those are fine.
Okay. And last question: when is the goal for this to be ready to be available for non-test phase use? So we're looking at up to age 17 now. When might it be available for general autistic public?
So we have to go through several steps here. And this is the sort of the classic at the end of the story. We say this is really encouraging, but you have to wait 20 years for the thing to be available. I hope that's not the case. What we're planning is to finish this study. If it reads out positively, we will go into phase 3, but that phase 3 will include 5 to 17-year-olds. And we have to go through that in order for FDA to approve it. And that will take at least another 3 or 4 years before we get through that and then get permission to sell it. In parallel with that, we would plan to start doing trials in adults as well, and with the goal of getting the data to expand the label for adult use after that. So we think it will still be a few years, but not like a decade, right? So I think we're kind of in that really interesting spot where you know, we can see the light at the end of the tunnel. We know what we have to do. So this is one of those I think encouraging ideas or therapies that actually I think we can say, you know, we plan to get this on the market in the next few years.
I assume the future studies will include an open label extension.
Absolutely.
Okay, now the people who will be in this study, I assume will be eligible to go into an open label extension when it's available.
Absolutely right, yes.
Okay, now what about the age issue though? If a 16 or 17-year-old goes into this study and finds that it helps, and then they're 19 when the open label study becomes available for the 13 to 17-year-olds, can they be grandfathered in?
Good question. We will have to get that. We would have to have that argument with FDA, I think, and discuss that with them. I would love to be able to include them, but I think the safe answer for me right now is to say they would not qualify. However, I think that's an open discussion for topic for FDA when we at our end of phase 2 meeting.
It's an issue of fairness.
Yes, absolutely. And the FDA, to their credit, has been very open-minded and very patient centric in all of our interactions. So they really know the community needs something like this. So I think they will be, like, obviously can't predict what they will do, but they've been very open to all these types of discussions, and we just have to present our case as strongly as we can. And we've started to engage a lot of advocacy groups now with the goal of just not making more than making ourselves, our company and our work aware, them aware of our work, but more to make sure we understand what the community needs and what they want and what's going to make a difference to them. And we want them to come with us to FDA and be advocates for this. So we're starting that now. A lot of companies wait until the very end and try to sort of wedge in some patient feedback. And I don't know how you do that with a good conscience in this population.
So thanks, Stu and Sarkis, and thank you to everyone for the participation.